genome evolution by double-stranded breakage. Double-strand breaks in which both strands in the double helix are damaged. DSB is the abbreviation for double-stranded break. Double-stranded break can lead to genome evolution or the genome rearrangements. So double-stranded break is such a breakage in the DNA where both strands of double helix are damaged. There are three different mechanisms which are used to repair the double-stranded breaks. Those mechanisms, they are non-homologous end joining. Second method is microhomology mediated end joining, and the third method is homologous recombination. So there are three different methods by which double-stranded breakage repairs. In case of mammalian cells, double-stranded breakage in cells are primarily repaired by two methods. First method is by homologous recombination, and the second method is non-homologous end joinings. So these two methods are used to repair the double-stranded break in case of mammalian cells. Non-homologous end joining pathway does not require homologous sequence. In case of homologous recombination, there should be the homologous sequence, but in case of non-homologous end joining, there is no need of homologous sequences. Repair mechanism which is present in the cells or in the genomes of the mammals and the eukaryotes, that repair mechanism repair most breaks correctly, but sometimes it is able to induce the translocations in the chromosomes that can be in the form of chromosome rearrangements. These type of the translocations or chromosomal rearrangements, they are actually the basis in change in the genome, and they are basis for the genome evolution. In this diagram, we can see that there is a double-stranded break in the DNA. This double-stranded break can be repaired with the help of homologous recombination. These are the homologous chromosome, and they exchange their parts, and these breaks, they are repaired. Here we can see that both strands, they are broken. So these both strands, they can be repaired with the help of homologous recombination, but the double-stranded break repair can be with the help of end joinings. So end joining, they are of two types. One is called as microhomology mediated end joinings, and the second technique is called as non-homologous end joinings. So double-stranded break can be repaired by three different methods. One of the method is homologous recombination, second is non-homologous end joining, and the third is microhomology mediated end joinings. This is another diagram in which we can see that how double-stranded break are repaired. Here we can see there is a double-stranded break. This strand is broken and this strand is also broken. So when there is a double-stranded break, then Ku complex binds to the DNA here. Then MRX complex is recruited, this one. Then DNA ligase complex is recruited. And at the next step, DNA polymerase and RAD27 nuclease arrive, and the lesion is repaired here. So this is the mechanism that how double-stranded break, they are repaired within the cells. These are the key players which help or which perform different functions to repair the double-stranded break in the DNA. So if we conclude that what is double-stranded break? Double-stranded break is actually double-stranded break in both strands of the double helix. As the DNA is double helix, there are two strands of the DNA. If both strands of the DNA, they are broken, such a break is called as double-stranded break. So double-stranded break can lead to genome evolution, and they also lead to chromosomal rearrangements.